Yes. Yes. Very good morning, dear students. So today I'm going to start with a new topic called microbes in human welfare. A very much important, very one of the chapter which is going to contain some 2 to 3 questions minimum for NEET examinations. So microbes in human welfare, the most important topic as far as class 12 topic is concerned. So इस chapter में हम क्या पढ़ेंगे? Microbes in human welfare में we are going to talk about the different types of microbes which in our day-to-day life they are very much beneficial for human mankind. Okay, for humanity. And to start off with, the best one in this chapter, microbes. So when we talk about microbes, let us start with the definition of microbe. What is the definition of microbe? So they are microscopic in structure. They are microscopic. So they are microscopic, which you can see by the help of compound microscope, and they are bacteria. They can be yeast, a unicellular organism. They can be fungi. They can be some, but not of human use. Protozoa. So these are all microbes which you can see under compound microscope or in a compound microscope. So these are microbes, compound microscopes. So the organisms that you can see under microscope, and they are microbes. And the topic for today is microbes in human welfare. So what is the topic? The topic is about microbes in human welfare. So when we talk about the microbes in human welfare, what comes in our mind? If you see the NCERT book, what are the major topics being given? Tushar, one by one. The major topic. First, household product. So the first one is microbes which are utilized in household products. The second one is what? Industrial application. The microbes which are used in industrial applications. That comes next. Third is sewage treatment. That means we are going to talk about STP, sewage treatment plant, sewage treatment. Then biogas production. Fine. Biogas production. And the last one is, I think, biofertilizers and biocontrol agents, I think. Biofertilizers and biocontrol agents.
Sir, what's your mind?
No. Online class going on. Biofertilizers and biocontrol agents. Biofertilizer and biocontrol agents. I think this is the last topic. So today we are going to talk about the first one as household product. So under household product, what are most important aspects being given in your book is lab. Number one is lab. So let's talk about lab. What is lab? Lab is lactic acid bacteria. So when we talk about lab is lactic acid bacteria. Now, this lactic acid bacteria is normally what comes in our mind immediately when we talk about lab. What comes in our mind is about lactobacillus. Now, take care if phone comes. Lactic acid bacteria. Now, lactic acid bacteria are the group of bacteria. They are the group of bacteria which can, group of bacteria, which can convert milk into curd, which can convert milk into curd. বুঝিয়ে বলেছিস? Why I am not being able to? Yes. Milk to curd. So group of bacteria which can convert milk into curd. Okay. So what is milk basically? Milk basically is a type of emulsion. That means it contains what? Two main compositions. Tell me. Milk contains protein. What is the name of the protein? Casein is the protein. Milk contains protein, casein. It contains fat. It contains water. It contains water. It contains water. Now, what happens? Curd means what? Curdling of milk. That means when the milk coagulates, it turns into curd. When the milk coagulates, it turns into curd. So what is that? What is that? See, these bacteria, basically what happens? The protein present inside the milk, it basically binds the fats with water. Fats with water. Now, this protein, it gets denatured at some temperature. So for curd making, it needs what kind of temperature? In your household activity, when your mother changes milk into curd, she keeps this curd in ambient temperature. What is the temperature? Do you think if she keeps this curd after introducing the inoculum? I think this is very, very important. This is there in your NCERT. Inoculum. What is inoculum? A small amount of curd being taken. A small amount of curd being taken as inoculum because it contains billions of bacteria. It contains billions of bacteria, and then this curd, this inoculum is inserted in the particular milk, and the milk is heated a bit. And then it's being stirred. And then she's going to keep where? She's going to keep this curd or would-be curd where? In what kind of temperature? In low temperature or in ambient temperature? Come on, think about it. Optimum temperature is required for the activity of this bacteria. So what are the bacteria? Lab stands for lactobacillus. Lab stands for lactobacillus. Lacti. Lactobacillus lacti. Fine, lactobacillus lacti. So this is the name of the bacteria. It's one of the bacteria which can make this curd. Now, so it depends upon the inoculum, what kind of inoculum is being given and what kind of temperature is being maintained. On these two factors, the type of curd that you will be getting, it depends upon the two things. One is optimum temperature and the type of inoculum. Got the point? Got the point? If you buy a particular curd, like, which is already packed, are you going to take this inoculum and make curd in your home? Think about it. No. And tell me why. Because that is what has happened with that. What has happened with that particular curd? That particular curd is already processed. Fine? Now, so this is lab. So what are the most important thing that we need to study according to your NCERT? So when we talk about household product, microorganisms like lactobacillus and lab grow in milk and convert into curd. During growth, the lab produces acid. So what the lab produces, what is this bacteria produces? The bacteria produces acid. Fine. Think about it again. Let me just give you this idea again. In infants, when they, there is an enzyme in their stomach and called renin, and in the acid present inside the infant's stomach can change that milk. They take milk, and when they vomit, you have seen curdling occurs. The milk gets coagulated. The milk gets coagulated. And what is the science behind it? The science behind it, acid is going to change this particular milk into curd. So lactobacillus acidophilus, lactobacillus lacti, what they can make is they can change this particular medium acidic, and that's why it can help in curdling. That's why it can make into curd. During growth, the lab produce acids that coagulates and partially digest the milk protein. So what is the milk protein? Shojit, what is the milk protein? The milk protein is casein. The milk protein is casein. A small amount of curd added to the fresh milk as inoculum. That's very, very important, which is also called as starter. So this is also called as starter. Lab also improves its nutritional quality by increasing vitamin B12. So what is the function of lab? Lab can increase the vitamin B12 quantity in the particular curd, and also it can check the growth of different types of disease in your gut. In our stomach too, the lab plays a very beneficial role in checking. It prevents the growth of different types of other bacteria, harmful bacteria. So curd is good for you, bad for you? Come on. Curd is good for you. Clear? Chalo. So this is about the first household product. What is the second household product? And that is dosa and idli. It's a fermented product, we know. Dosa and idli. What is being changed? Dough, which is used for making dosa and idli, is fermented by bacteria. So when we talk about fermentation, if you remember, or if you have seen the video of respiration in plants, I've talked about fermentation. Let me give you a basic idea about that, and then we are going to get into idli and dosa. Okay? Idli and dosa. Kide bachcha naki? Idli and dosa. Like dosa ball, like bung diye etani exercise guruche. That ball dosa. Yes. Asche. Rupata bolbe na. Okay. Ki bolte gechilam. Fermentation. What is called fermentation? In respiration in plants, we have talked about what is fermentation. The fermentation is what? Glucose changes into what? Pyruvic acid. How many carbon? 3 carbon. It is what? CH3, COOH. This is pyruvic acid. And this pyruvic acid gets changed into lactic acid. How many carbon lactic acid contains? 3 carbon. This is also fermentation. So over here, what is required? This NADH2 gets changed into NAD+. So what happens over here? Oxidation occurs. And this pyruvic acid is being reduced. This pyruvic acid is reduced to lactic acid. But over here, you can see 3 carbon remains as a 3 carbon. So there is no liberation of carbon dioxide. There is no liberation of carbon dioxide. So carbon dioxide is not being produced during this particular fermentation. But this fermentation takes place where? In which part of the body? Come on. Muscles. Very good. It takes place in which part of the body? It takes place inside muscles, and even in yeast cell. It can occur. But from pyruvic acid, there is another one which can form into ethanol. From pyruvic acid, it can also form into ethanol. But it's not going to be able to make straight away ethanol. First, it has to make acetaldehyde, CH3CHO, which is acetaldehyde. Now, for making acetaldehyde, you see what has happened in making acetaldehyde. There is CH3CH. How many carbon it is having? 3 carbon. Acetaldehyde is having how many carbon? 2 carbon. So definitely what has happened? Decarboxylation has occurred. And this is done by what kind of enzyme? No. Pyruvate decarboxylase. Decarboxylase. Pyruvate decarboxylase. Okay. This acetaldehyde now changes into CH3CH2OH. So what has happened over here? Reduction. And for this reduction again, what has been formed? For this reduction again, this has been reduced, but this NADH2 has been oxidized to NAD+. And normally we know from glucose to pyruvic acid, what is the net gain of ATP? 2 ATP. How? 4 ATP is formed directly by substrate level phosphorylation and 2 is being utilized. So what is the net gain? 2 ATP. So during fermentation, what is the number of ATP we can get by this whole fermentation? 2 ATP. Any issues with this? Now, what is the name of this enzyme? What is the name of this enzyme? The name enzyme is, yes, alcohol dehydrogenase. Alcohol, because what is this process? Oxidation. And what is this process? Reduction. Reduction and oxidation taking place simultaneously. We call it oxidoreductase enzymes activity, and that is being done by dehydrogenase enzyme. So alcohol dehydrogenase is the name of the enzyme. Alcohol dehydrogenase is the name of the enzyme. What is the name of this enzyme? This is lactate or pyruvate? Pyruvic acid to lactic acid. It should be pyruvate dehydrogenase. Pyruvate dehydrogenase. Dehydrogenase. Clear? So ये था fermentation आप लोगों के सामने. तो देखो यहाँ पे क्या निकला? Carbon acid निकला. And this takes place where? ये क्या बना? Ethanol. What is this? This is ethanol. That means it's an alcohol. It's an alcohol. So this alcohol making cell is what? Is yeast. Is yeast, unicellular fungus. And this yeast, you know the name of this yeast as Saccharomyces cerevisiae. Saccharomyces cerevisiae. And this Saccharomyces cerevisiae can be called as two types of yeast. It can be either called as brewer's yeast. In brewer's yeast is going to make what? Beverages. And what is the beverage? Alcohol is the beverage. And during alcohol formation, what is being released? Carbon dioxide is released? Yes. But another one is called baker's yeast. Baker's yeast. Baker's yeast. Now, baker's yeast is going to make what? Bread. In bread making, there is a puffiness which occurs. Come on. There is a puffy appearance. Puffy appearance. That puffy appearance is due to the release of what? Carbon dioxide. It is due to the release of carbon dioxide that there is a puffy appearance. And this puffy appearance we have also seen in cheese. So cheese is also a fermented product. Cheese is also. And what is the best animation you have seen cheese in? Tom and Jerry. Clear? The moment Jerry opens the refrigerator, watches that particular cheese, eyes comes out as balls. And then there is a large sized hole. So he's going to reach inside the hole and, allure Tom, whatever. There is a Swiss cheese. What is a large hole made up of? Carbon dioxide. So puffiness. But one thing is different. My question to you, just tell me one thing. In brewer's yeast and if in baker's yeast you compare, fermentation means it should take place in presence of oxygen or in absence of oxygen or in both. In both. Fermentation, in case of baker's yeast, can take place in aerobic and anaerobic. Got the point? But for brewer's yeast, the prerequisite condition required is completely anaerobic. Got the point? And that is why it can lead into formation of carbon dioxide and ethanol. Clear? Any issues with this? So it's very, very important. Baker's yeast, aerobic and anaerobic conditions, and ultimately it forms the puffy appearance. So this is about yeast which can make beverage, alcohol, and bread making. So this is also used in a household activity. And also on a large scale, beverages can be made in industries. In large scale, beverages can be also made in, alcohol can be made in large scale in industries. In your book, you can see that fermenters. There's a figure now called fermenters. Good. So this is about yeast. Now, I was talking about fermentation and lactic acid. Dosa, no? I was talking about dosa. Idli and dosa. Dosa, in English. It should be dosa. Dosa and idli. In both of them, there is a puffy appearance or not? There's a little bit of puffy appearance. So what is released? Fermentation occurs. Fermentation occurs. And ultimately forms into what? Forms into, it releases carbon dioxide. Releases carbon dioxide. Okay. Let us come back to the NCERT. So dough, which is used for making dosa and idli, is fermented by bacteria. The puffed up appearance of dough is due to the production of CO2 gas. Clear? So the puffed up appearance is due to the production of carbon dioxide gas. Dough, which is used for making bread, is fermented by fungi, Saccharomyces cerevisiae. It's called as a breaker's yeast. There's another one traditional drink. There's another traditional drink in southern India, and that is toddy. That is toddy. In Bengali, it is called as tadi. In Bengali, it is called as tadi. Fine. So this is the palm one, palm tree. Okay, palm tree. You have seen certain particular pot is kept over here throughout the night to get the sap of the palm. So this is palm tree, and the sap of palm tree gets fermented. Because the sap contains glucose and fermentation occurs, and this fermentation ultimately helps in the formation of what? It helps in the formation of what? Alcohol up to 13% to 14%. Alcohol up to 13 to 14% of alcohol is being produced by this. Alcohol is being produced by this. Clear? Okay, so this is toddy, traditional drink. In Bengali, it is called as tadi. Tadi, in Bengali, it is called as tadi. Fine. It's a fermented product from the sap of palm. Any issues? Clear? Okay. Cheese. Cheese is one of the oldest food items in which microbes were used. Different varieties. Cheese. Cheese. Cheese is one of the oldest food items in which microbes were used. Different varieties of cheese are known by their characteristic texture. See? Cheese are known for their characteristic texture, flavor and taste, and specificity coming from the microbes used. So cheese depends upon what kind of microbes is used. Acha, what is cheese basically? Come on, what kind of information you have got about cheese? Tell me. चीज खाते हो आप लोग, बोलो चीज के बारे में आप क्या जानते हो? What is cheese? Cheese is a milk product or not? Yes, it's a milk product. It's a milk product. So fermentation करने के बाद चीज में fermentation हुआ, fermentation करने के बाद ultimately क्या बनेगा, क्या इसके ऊपर act करेगा, what is going to act upon this milk? So milk is having the protein, और वो उसमें क्या किया जाता है मिल्क के अंदर, if you add vitamin C, if you add citric acid, it's going to get what? The water and the milk product is going to get separated. And on that separated product, if you add a bacteria named Propionic bacterium Shermani and double I, Shermani, वो शर्म से बनाता है क्या? चीज. Propionibacterium shermani. Now let me tell you one thing. This particular bacteria can to some extent make a component called propionic acid, which is also a fermented product, which is not there in your book. And propionic acid. And this Propionic shermani ultimately makes what? That Swiss cheese, which is having large hole. Swiss cheese, which is having large hole, that is Tom and Jerry, fine. Tom and Jerry, special, Swiss cheese. Now in this cheese is produced by the bacteria, it's a fermented product of milk. But what gives the characteristics flavor or texture or taste of it? If this particular cheese is infested by one kind of bacteria which is Propionibacterium shermani, and also it can be infested by some other fungus for some taste or flavor. Now in class 11 under the chapter classification, when I have taught you fungi, when I have taught you fungus, I've talked about a type of Ascomycetes named penicillin. Penicillium notatum. Penicillium notatum makes what? Penicillin as an antibiotic. We are going to study today also under industrial usage. Fine. We have talked about one kind of, if this cheese produced by Shermani, the cheese produced by Shermani is infested by a special type of fungus named Penicillium roqueforti. Penicillium roqueforti. Then this cheese becomes Roquefort cheese. This cheese becomes Roquefort cheese. So, on what basis the cheese depends upon? First, on the type of organisms it is infested by. Clear? There is another type of cheese. Tushar, tell me. I have talked about another type of cheese and starts with the letter C. C for cheese? No. C for Camembert cheese. And this Camembert cheese is produced by the infestation of another type of fungus named Penicillium camemberti. Penicillium camemberti. This is the name of the cheese. Penicillium camemberti is the name of the fungus which can make camembert cheese. Any issues with this? Come on. Great. So this is about the cheese. Okay. Now cheese is a microbe, it is produced by the microbes. Now, after cheese, Roquefort cheese are ripened by, large holes in Swiss cheese are due to the production of large amount of carbon dioxide by bacterium named Propionibacterium shermani. It came 2019, NEET. Roquefort cheese are ripened by growing a specific fungus on them, which gives them a particular flavor. We have talked about another one called Camembert cheese. Now let's get into microbes in industrial products. When we talk about the microbes in industrial products, we are going to talk about these are fermenters, large fermenters in your NCERT, and these are fermentation plants. These are fermentation plant you can see in the NCERT. Fermented beverages, just now we have talked about that. Fermented beverages, what are these? Microbes, mainly yeasts. Used for production of beverages like wine, beer, whiskey, brandy, rum. Now these three, whiskey, brandy and rum, these three are distilled. These three are distilled. So they are produced by distillation. But wine and beer are non-distilled. They are without distillation. Wine and beer are produced without distillation. Clear? Now, for this purpose, the same yeast, Saccharomyces cerevisiae, used for fermenting malted cereals and juices to produce ethanol and commonly called brewer's yeast. So malted, what is being used? The grains is being used, barley, wheat is being used, malting process is being used. Can you tell me the name of a hormone, plant hormone, which is used for malting or brewing? Gibberellin, gibberellic acid. Okay, so this is brewer's yeast. Depending upon type of raw material used for fermentation and type of processing, different types of alcoholic drinks are obtained. Now you know Sometimes people, they take the rice and they dip it inside water and keep it like that, allow it to ferment, and they make that particular alcohol. Fine. So that is also it. So it depends upon what kind of raw material you are using. You can make this kind of alcohol from different types of grapes. You extract the juice of the grapes and then put yeast over there. You can get the alcohol. So on what kind of alcohol is going to form depends upon what kind of raw material is being used. What kind of raw material is being used. Whiskey, brandy and rum are produced by distillation. Whiskey, brandy and rum are produced by distillation. That is the most important. And without distillation is wine and beer. Wine and beer. Okay. Sure. Antibiotics. So this was about the first industrial product that we have made in a broad, large scale, that is beverages. Now let's come into antibiotic. So let us start with the antibiotic. Let us give you some of the idea about antibiotic first, and then we are going to move into the what NCERT has got to say. When we talk about antibiotics, what is the meaning of the term anti? Anti means against. Against bio, against life. It's against life. That means it is not going to allow any life to form in. So antibiotics, when we talk about antibiotics, this is the name which comes in our mind. Alexander Fleming. He was culturing a type of bacteria called Staphylococcus aureus. I think you know what is called Staphylococcus. Coccus means what? What kind of bacteria? Staphylococcus. Coccus means what? Oval shaped. So this is the name of the bacteria called Staphylococcus aureus. So he was culturing this bacteria in a nutrient medium, and suddenly found that in this master plate, some portion of this bacteria were not growing. And when he found and had a close watch under microscope, found some of the thread-like structure were growing over there. So the thread-like structure were secreting some of the chemicals, and those chemicals acted as antibiosis. Those chemicals acted as antibiosis. And what is antibiosis? Against life. So this kind of thread-like appearance structures, which are called penicillium, Penicillium notatum. So it was discovered that this kind of spores which has come over here. Who could tell me what is the name of the spore of Penicillium notatum? Conidiospore. Fine. So this conidiospore ultimately fell off from some of the areas, and ultimately, they started growing. And when they were growing, they started secreting this kind of antibiosis. It's a chemical. And this chemical is never going to allow the bacteria to grow. So this part of the region where fungus were growing, this part of this region, the bacteria were not growing. They stopped growing. And this kind of relationship between fungi and bacteria, is what? Bacteria is getting killed by the presence of fungi. So for fungi, it is 0. And for bacteria, it is negative for them. So this kind of symbiotic, this kind of association is called, amensalism. We are going to study the same in organism and population. Amensalism, where for fungus, they are neither being harmed nor being benefited by the presence of bacteria. But it is due to the fungus that bacteria are getting killed. Clear? So this is antibiotic. So Alexander Fleming accidentally got this penicillin, and this penicillin was widely used in World War II, and the usage was done by the help of 3 of the scientists. What is Ernst Chain, Howard Florey. So these 2 scientists, they utilized this antibiotic penicillin in World War II. in World War II. World War II, 1945. And they got Nobel Prize. They got Nobel Prize along with whom? Alexander Fleming. So these three scientists, they got Nobel Prize. Alexander Fleming. It's there in your NCERT. It's a very, very important aspect. Einstein, Howard Florey, and Alexander Fleming. Let's find out. Antibodies produced by microbes are regarded as one of the most significant discovery of 20th century. Now see, in any war, if there has been a slight cut due to any bullet, or see, there has been the whole legs has been amputated. So if there has been a whole leg has been amputated due to the blast of the landmine, the soldiers used to die, because if there is a small cut, the first line of defense has been ruptured. So bacteria can easily get inside your body through that small portion of your skin where there is no keratin, because keratin protects the skin from the invasion of the bacteria. And there has been a large cut, the whole leg has been amputated. So the person would have died by the condition called septicemia. So antibiotic was the greatest discovery of 20th century, but because by injecting this antibiotic, the person might get saved. Now, antibiotics are chemical substances produced by some microbes and can kill or retard the growth of other disease-causing microbes. ये क्या कर सकता है? Other disease-causing microbes को वो मार सकता है. कौन? Antibiotic. The first antibiotic discovered was penicillin, and it was a chance discovery. Majority of discovery are chance discovery. Alexander Fleming, while working on Staphylococcus aureus, it's not been given, Staphylococci bacteria, once observed a mold means fungus, growing in one of his unwashed culture plates around which Staphylococci could not grow. Got the point? Any issues with this? Come on. Good. Fleming found that it was due to a chemical produced by mold, Penicillium notatum. Penicillium notatum. He named it penicillin. Full potential of penicillin as an effective antibiotic was established by Chain and Florey. The effectiveness of this antibiotic was discovered by Chain and Howard Florey and Einstein. Howard Florey and Einstein. Fleming, Chain and Florey were awarded the Nobel Prize in 1945 for this discovery. Penicillin was extensively used to treat American soldiers wounded in World War II. Used to treat, and that was a point of discussion. It was used only with American soldiers. Mind you, that was a point of discussion. Worldwide people, they oppose this particular phenomenon, this particular act. Antibiotics are used to treat deadly diseases such as plague. Who is going to tell me? Plague is caused by? Tell me the causative agent. Plague is caused by? Plague. Pasturella pestis. So antibiotics can be used against this bacteria, plague. Whooping cough, kali kasi, Bordetella pertussis, important. Bordetella pertussis, kali kasi, whooping cough. The other term for this disease is pertussis. Diphtheria, galghotu is important. The Hindi term is important. Diphtheria caused by Corynebacterium. Diphtheriae. Corynebacterium diphtheriae. Leprosy. Kushtrog. It is caused by leprosy. Mycobacterium leprae. It is caused by Mycobacterium leprae. Leprosy is caused by. So very, very important, the causative agent that you know. You have to Remember all this. Clear? So this is about antibiotics. So antibiotics are used to treat deadly diseases like this. Okay? Very good. Sure. Next. Chemical enzymes and other bioactive molecules. Whenever a teacher wants to make question from this chapter, what comes immediately in their mind is about this portion called chemical enzymes and other bioactive molecule. Is a favorite portion for them. ये बहुत important area है जहाँ से क्वेश्चन आप expect कर सकते हो. Fine, you can expect questions from here. Yeast, that is Saccharomyces cerevisiae, production of ethanol. We have just talked about that. Lipases, lipid को खाने वाला agent. Lipases. शादी बाड़ी में जाते हो, लाइट कलर का शर्ट पहन के, तो हो गया यहाँ पे मटन का जूस गिर गया यहाँ पे. मटन के जूस में है क्या? मटन मतलब ही फैट, प्रोटीन है, और आपको कौन सा स्पेशल लगता है? फैट. ओके, नाउ तो वो कैसे जाएगा? यूज इन डिटर्जेंट फार्मूला. लाइपेज डिटर्जेंट फार्मूला में है, फॉर्मुलेशन में है. So the detergent having lipases, the lipase is going to feed on the lipid, and that lipid को वो digest कर लेगा तो वहां से निकल जाएगा, and are helpful in removing oily stains. This is how oily stains are being removed from the clothes by adding lipases. So these are enzymes which are on a daily basis, on a day-to-day basis, used in your household activities. Clear? Sure. Next. Next is bottled juice. Very, very important. Bottled juice. कौन सा juice आपको पसंद होगा? The bottled juice having some fibers sedimented. अच्छा लगेगा आपको? एप्पल जूस नीचे कुछ फाइबर गिरा हुआ, आप गॉड ये क्या है, नहीं पिएंगे. So company, they have what they have used. They have used to make it clarified. They've used bottle juice में. They have used. See, these are very, very important questions for NEET. ये बहुत बार आ गए हैं. Proteases and pectinases. Pectinases किसको feed करेगा? Pectinases are going to feed upon the pectin. And what is pectin? It is a calcium pectate, the main composition of the middle lamella. Okay, they will feed on the pectate and also the protein part of the cell membrane. So what is going to happen? पूरा clarify हो जाएगा. Streptokinase enzyme, they act as a clot buster. It's produced from Streptococcus bacteria, and this streptokinase enzyme, ये जब आप डॉक्टर करोगे, इमरजेंसी ड्यूटी करोगे 36 घंटा का, तो जो इमरजेंसी पेशेंट आएगा आपके पास, आपको समझ में आ गया by watching on the symptoms that the particular patient is showing off, आप क्या इंजेक्ट करोगे? इफ यू थिंक कि वो क्लॉट रिमूव करना है, क्लॉट हो गया है, सो स्ट्रेप्टोकाइनेस इंजेक्शन आप अप्लाई करोगे. इट इज मॉडिफाइड बाय जेनेटिक इंजीनियरिंग एंड आर यूज्ड एज अ क्लॉट बस्टर, क्लॉट को निकालने वाला एजेंट, for removing clots from the blood vessels of patients who have undergone myocardial infarction. What is the other term for myocardial infarction? A heart attack. MI, myocardial infarction, leads to heart attack. Tell me the symptoms of myocardial infarction. The pain is going to, what is the pain in the heart? Called angina. And the pain is going to radiate towards your left arm, left chin, left shoulder. Anything done with the right side, gas. Clear? Yes. Left side, don't consider it to be gas. It's a problem of MI. Clear? Okay. So MI, myocardial infarction, streptokinase injection is a very, very important one, which burst of the clot if there has been any. Cyclosporin A, very, very important bioactive molecule, is produced by Trichoderma polysporum. Mind you that Trichoderma polysporum. We have seen this. We have found this Trichoderma in human health and disease topic. Trichoderma polysporum is a fungus which can produce a component called cyclosporin A, which is used as immunosuppressive drug. Immunosuppressive drug. Say for example, I have given my kidney to Sojit. Now throughout the life, Sudipto has to get cyclosporine A because his body is always going to try to reject my kidney because my kidney is antigen for his body. So the immune system of Sudipto's body has to be suppressed by the component called cyclosporine A, which is produced from Trichoderma polysporum, which is a fungus. Cyclosporine A is used as immunosuppressive drug agent in organ transplantation patient. Clear? Now, statins. Very, very important one. The next one is statins. So when we talk about statins, what is important about statins? Produced by Monascus purpureus. It's an yeast. What is yeast? Unicellular fungus. Monascus purpureus, statins. Statins used as blood cholesterol lowering agent. Fine. आपके घर में आपके पिताजी ये खाते हैं, भगवान न करे, पर अगर खाते हैं तो ये खाते हैं एटोरवास्टाटिन, रोजुवास्टाटिन, लास्ट में सफिक्स में स्टैटिन रहेगा, मतलब क्या है? It's a blood cholesterol lowering agent produced from the organism Monascus purpureus. Clear? Very good. Now, chemicals, enzymes and bioactive molecules. Static acid कहां से बनेगा? लास्ट स्केल में देखो, by Aspergillus niger fungus. Aspergillus niger. मुझे बोलो, Aspergillus niger comes under which class of fungus? There are 4 classes of fungus. Tell me the name of the fungus classes. Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Tell me Aspergillus niger comes under which class of fungus? Ascomycetes. Sir, second term. PABD. Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. Second term on Ascomycetes. Okay. Acetic acid is in large scale produced by Acinetobacter acidi, which is a bacterium. On a large scale, Acinetobacter acidi, large scale acetic acid being produced. So it's also a very, very, very important chemical. Butyric acid, Clostridium butylicum. It's a bacterium. See, 2016, 20, 24. 4 साल. Next one 2018 में आएगा, 2028 में आएगा. 13, 16, 20. 3, 4, 5. Okay, so it's very, very important. So this bioactive molecule is very, very important one. So you should not miss any of the questions from here. Now, Clostridium butylicum. Tell me any other information you got about Clostridium butylicum. I've told you about it. No.
Oh,
Jute, very good. Rating of fibers. পাট গাছ কেটে পুকুরে ফেলে দেওয়া হয় পচানোর জন্য। ওই পুকুরে কি অ্যাসিড তৈরি হয়? বিউটারিক অ্যাসিড। কোনদিন মাছ চাষ হবে ওই পুকুরে? হবে না। Now what happens for rating of fibers, the jute formation? What happens? This kind of bacteria, they feed on the other part and makes that the antacid part is the jute. So rating of fibers is done by Clostridium butylicum. It's very, very important. Rating of fibers. I'm writing it over here. Rating of fiber. Rating. RETTING. Rating of fibers is done by Clostridium butylicum. Lactic acid formation by Lactobacillus bacterium. You know that Lactobacillus lactic acid. Ethanol, Saccharomyces cerevisiae, yeast. Streptokinase, Streptococcus bacterium. Cyclosporine, Trichoderma polysporum. Statin, Monascus purpureus. Just see the year. See how many times this question has been repeated. How many times the question has been repeated from this topic. So this particular topic under NCERT, chemical, enzymes and bioactive molecule. I think old book, page number 180. New book, page number 153. Page 183, old book. Clear? Very, very important. This chapter, especially this page. Microbes in sewage treatment. थोड़ा बाद में देखेंगे इसको, ये बड़ा टॉपिक है. Hopefully next day we are going to start with this one. थोड़ा हम microbes in production of biogas को देख लेते हैं. Production of biogas. Production of biogas. Biogas matlab. We know that there is a bacteria named methanogen. Methanogens. Kahan pe mila hame ye term? Where from we have got this term? Earlier in class 11, we have studied in the chapter Archaebacteria under Monera phylum, under kingdom Monera. We have studied there are two types of bacteria. One is Archaebacteria, and the one is Eubacteria. Eubacteria, Archaebacteria and Eubacteria. Archaebacteria, there are three types of Archaebacteria that we have studied. One is halophiles, thermoacidophiles, and methanogens. Under methanogens, the example is Methanococcus. Methanococcus. And they are present in the ruminant's stomach. Ruminants. What are ruminants? What are ruminants? What are ruminants? Cow is having 4 chambers in stomach. 4 chambers in stomach. Fine. This is rumen. This is reticulum. This is abomasum. And this is omasum. Which one is a true stomach? What kind of stomach you are having? Abomasum. It's a true stomach. It is called as the true stomach. So these 4 chambers are present in majority of the herbivores, except, tell me the exception. Name the organism which is not a ruminant. Camel. So the food is going to come through esophagus, get inside rumen. Rumen contains a bacteria named Rumenococcus. Rumenococcus. And this Rumenococcus is a methanogen. That means it creates methane gas. And that is why the cow dung contains. The cow dung contains what? Methane. The cow dung contains methane. Okay, so let's comment over here. So in your book, they have given one of the example of this bacteria is Methanobacterium. Methanobacteria. We want to study that. So let us see. By fermentation of dough, cheese making and production of beverages, the main gas produced is carbon dioxide. Okay? Is carbon dioxide. Certain bacteria which grow anaerobically or on cellulosic material produce large amount of methane along with carbon dioxide and hydrogen. These bacteria are called methanogens. So what are the gases? Methane, carbon dioxide and hydrogen. And one such common bacterium is Methanobacterium. These bacteria are commonly found in the anaerobic sludge. What is sludge? We will talk about that in STP, sewage treatment plant. Anaerobic sludge during sewage treatment. These bacteria are also present in the rumen. Rumen, a part of the stomach of cattle. What is the name of the bacteria I talked about right now? Rumenococcus. A lot of cellulosic material present in the food of cattle. So, what material? Cellulosic material. They feed on what? They feed on meat or plant products. So, plant product contains cellulose. What kind of material is cellulose? Homopolysaccharide or heteropolysaccharide? Homopolysaccharide. Present in the rumen. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. Any issues? Come on. Any problem? Thus gobar, excreta dung of cattle is rich in these bacteria. Dung can be used for generation of biogas, commonly called as gobar gas. Biogas contains, star mark, biogas contains methane 70%. Very, very important. I think this information is not there in your NCERT. Biogas contains 70% of methane, 25% of carbon dioxide, 1% and 1% of hydrogen sulfide and hydrogen. So 70, 25 and 1. 70, 25 and 1. Methane 70%, carbon dioxide 25%, 1% of hydrogen and hydrogen sulfide. The technology of biogas production was developed in India mainly due to the efforts of IARI, Indian Agricultural Research Institute, Indian Agriculture Research Institute, and KVIC, Khadi and Village Industry Commissions. KVIC, Khadi and Village Industries Commissions, KVIC. Got the point? How it looks like? How it looks like? This is how. What kind of questions can you expect from this diagram? Diagram will be given and labeling questions can come down. See, dung water. So this is the raw material, dung water. Human excreta is not going to function over here. Pushpita, what is the reason? Human excreta is not going to work. Human excreta দিয়ে biogas হবে? হবে, God, হবে না. Why? Because human excreta is not having, human stomach is not having rumen. It's having only abomasum. It's a true stomach. So rumenococcus bacteria is not there in your stomach. So dung water is the raw material. Dung water is going to go over here, digested by digester, and it is going to create methane 70%, carbon dioxide 5%, and hydrogen sulfide 1%. And this is going to go to your kitchen. Cock is there, and then ultimately gas. What is going to happen after some time? There won't be any other gas left. So it's going to come down as a sludge. Use this sludge in your as a compost. Use sludge as a compost and use it in making the soil fertile. Got the point? So this is about biogas plant. So Khadi Village Industries Commissions, what they give subsidy to the people who wants to make biogas plant. Say for example, biogas plant बनाने के लिए 50,000 रुपए खर्चा होगा. So 25,000 will be paid by the government when you make half of it, and the officials will come down and see. They're going to promote you to go for biogas plant formation. Clear? Because गांव में क्या raw material अभाव रहेगा? ये वाला अभाव रहेगा dung? No. So that was all about biogas plant. Microbes as biocontrol agent. I think we should not start with this. STP और ये दोनों next दिन complete करेंगे. चलो questions कर लेते हैं. Fine. Let us solve some questions. Chalo. First question. Raise your hand who can make it. Don't just answer me. Chalo. First question. Those who can make it, raise your hand. Tell me. Which of the following microbes is not involved in the preparation of household products? Not involved. Not involved. Question टेढ़ा है पर अपना है. बोलो. Not involved. Household product. Kire, tell me. B. Not involved. Oh, options. Options. चलो B, A and C. Aspergillus niger and C Trichoderma polystyrum. ठीक है? A and B नहीं होगा. अच्छा. C तो नहीं होगा confirm. Trichoderma polystyrum कहाँ से क्या करते हैं ये? Trichoderma polystyrum makes what? Cyclosporin A. Immunosuppressive drug. हर दिन किडनी ट्रांसप्लांटेशन होता है तो घर में हर दिन किडनी ट्रांसप्लांटेशन होता है नहीं होता है? सो नहीं होता है. So household product এটা यूज হয় না. Very good. So answer is B, A and C. And what about Aspergillus niger? What is the function of niger? It makes what? Citric acid. On a large scale? Okay. Chalo. Choose the correct answer from the options. Raise your hands. Pushpita, try this. Choose the correct answer. अरे कौन सा? अरे क्या किया? अरे चलो conversion of milk to curd. Vitamin B12. Vitamin B12. अच्छा vitamin B12 का chemical name बोलो. Tell me the chemical name of vitamin B12. Chemical name of vitamin B12. Cyanocobalamin. सायानोकोबालामीन। विटामिन डी बोलो। कैल्सीफेरॉल, कैल्सीफेरॉल, विटामिन डी। विटामिन D3 बोलो। कोलेकैल्सीफेरॉल, D3, कोलेकैल्सीफेरॉल। ए बोलो की रे, ई, हाँ ई बोलो, ई वृक्षा, ई विटामिन ई आप लगाते हो मुंह में, विटामिन ई कैप्सूल इवियन, हेल्दी हेल्दी बच्चों वाला स्किन, विटामिन ई, टोकोफेरॉल, टोको। So what is the answer? Curd increases the amount of vitamin B12. Is this clear? Hey, is this clear, Baba? Okay. Now, yes, the one who can answer me, raise your hands. Probiotics are. Oh God, I must have talked about this. I think you know what is probiotic. हाँ, बाजार में मिलता है याकुल्ट। हाँ, प्रोबायोटिक, प्रो मतलब टूवर्ड्स, probiotic. So probiotic, so which is good for your body, good for your gut. Probiotics, it contains some of the good bacteria. Live microbial food supplements, live microbial probiotics, Yakult. चल, डो बोलो। डो kept overnight in warm weather become soft and spongy because of, attraction the है अच्छा ना। Okay, बोलो। Dough kept overnight in warm weather becomes soft and spongy. Why not in cold weather? Not why inside refrigerator? How na? Yes, ambient temperature is required. I told you optimum temperature is required because of fermentation. Because of fermentation. Chalo. Which of the following is an example of non-distilled alcoholic beverage produced by yeast? Non-distilled. C, beer is a non-distilled. Wine and beer, both of them are non-distilled. Brandy, whiskey and rum are distilled one. Streptokinase produced by bacterium. Streptococcus is used for. Pushpita, tell me. Streptokinase. Very good. Removing clots from blood vessels. Removing clots from blood vessels. So they act as a clot buster. They act as a clot buster. Very good. Given below are two statements. Wait. This is very, very important. Check it out. Who can make it? Raise your hands. This is assertion reason. Assertion reason? No. Statements only. Chalo. Antibiotics are chemicals produced by microbes that kill other microbes. ठीक है? Antibodies are chemicals formed in body that eliminate microbes. Okay, eliminate microbes. Statement 1 is correct, both statement, but statement 2 is incorrect. Which one is the answer? Huh? C. Both statement 1 and 2 are correct. Acha. Antibodies are chemicals for. Acha, for statement 1 is right or wrong? Statement 1 is right or wrong? Correct or incorrect? Sure. Antibiotics are chemicals produced by a microbe. What is a microbe? Fungus is a microbe. The name of the fungus is Penicillium notatum. That kills other microbes. That is okay. That kills bacteria. Bacteria is also a microbe. Antibodies are chemicals formed in body that eliminate microbes. Is this statement right or wrong? And that is going to help you to answer it. For Pushpita, this statement is right. For Tushar, tell me faster. 45 seconds will be given in your exam. Chalo. To incorrect. Correct kya hobe? Can you incorrect? Ki dekhi incorrect? Antibodies are chemicals formed in body that eliminate microbes. Huh? Kill microbes of it. Antibodies kill microbes. Or antibodies binds with the antigen and make it ineffective. Antigen-antibody reaction. In antibodies, you have seen antigen binding site. In the Y-shaped structure, you have seen the antigen binding site. So antibody binds with the antigen and makes it ineffective. Makes it? What has happened? Okay. Yes. Tell me. A is correct. Eliminate microbes. Do you think the term eliminate microbes is okay? It eliminates microbes or it inactivates microbes? Answer, statement 1 is correct but statement 2 is incorrect. What is the answer? Which one? A. A is the answer. Very close call. Very close call. Tell me this. अच्छा এটা কি আছে? अच्छा चलो. हैं? मैच ऑप्शन कहाँ पे है? नहीं ऑप्शन. अच्छा बोलो आप फिर. अच्छा है. आप लोगों के लिए इजी हो गया. बोलो. क्लॉस्ट्रिडियम ब्यूटाइलिकम. ब्यूटाइलिकम. ब्यूटाइलिक एसिड कहाँ पे है? ब्यूटाइलिकम से ब्यूटाइलिक एसिड. ब्यूटाइलिकम से ब्यूटाइलिक एसिड. सैकरामेजिस डेरिवेसी. इथेनॉल. ब्रीवर्स ईस्ट. ट्राइकोडर्मा पॉलीस्फेरम. साइक्लोस्पोइन ए. स्ट्रेप्टोकोकस. स्ट्रेप्टोकाइनेस. चलो करो इसको. Identify the macroorganisms which is responsible for the production of an immunosuppressive molecule, cyclosporin A. D. Trichoderma polysperm. चलो करो इसको. Raise your hands who can do it. Faster. वही जल्दी faster. सोजित बोलो. এত ফাস্ট বলবি? আচ্ছা দেখ তোর आंसर কি? তোর आंसर কি? রূপকথার आंसर কি? বি. সোজিত এত ফাস্ট করিস না রকি কি ভুল ভুল হয়ে গেলে. আচ্ছা ঠিক है आप लोगों का इफेक्टिविटी ज्यादा है. आई नो दैट you are young, you have got good amount of what concentration power which is a little bit of lesser inside. Aspergillus niger is going to make what? C. Ascorbic acid make what? Acetic acid. Clostridium botulinum makes what? Butyric acid. Lactobacillus makes what? Lactic acid. Answer is B3, B1, B1 একটাই আছে. B1 good हो गया. 4-2 C4 हो गया. Very good. Very good. Match the following columns and select the correct option. Chalo. Wait. I want the answer from Rupkatha. Option A, A2, 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 A2. আচ্ছা A2 একটাই আছে, এইটাই গন্ডগোল করেছে, বাজে অপশন। এটা কোন ইয়ারের? ওটা 2020 ছিল এটা। এটা দেখছি তাই, মানে ধরে দিয়ে কাজ করে দিতে হবে। তাও একটু দেখিস, হ্যাঁ এখানে করছিস ওখানে একটু সময় দিস, সম্মান দিস একটুখানি। Clostridium botulicum, butylicum butyric acid, Trichoderma cyclosporin, Monascus perfidius, blood cholesterol lowering agent, statin, Aspirants niger, citric acid. So next, match the following organisms. Pushpita is going to tell me with the products that they produce. Lactobacillus, curd, Cerevisiae, bread, niger, citric acid, acidic. Oh God, কি হলো? Acidic? गंडगोल है, ऑप्शन गलत है। ই হতে পারে, ই হতে পারে না, না দুটোই তো माइनस হয়েছে, সো द ऑप्शंस गलत है। एस्पार्टिक एसिड हेल्प्स इन एसिडिक एसिड, बदले नहीं। एस्पार्टिक नाइगर साइट्रिक एसिड ठीक है, चीज नहीं है, चीज के लिए क्या होना चाहिए था? आइदर रॉकफोर्ट या प्रोपिनोमेटम सरमानी। ओके पुष्पिता बच गए आप। Which of the following is a commercial blood cholesterol lowering agent? You know that? Blood cholesterol, statin, blood cholesterol lowering agent, statin. Which of the following is currently matched for the product produced by them? Ball, এটা বল, পুষ্পিতা বল। Currently matched for me for the product produced by them. Ball, Pushpita ball. Currently matched. This is not currently matched. Not currently matched. Currently matched. Not currently matched answers. चलो चलो ओके। ट्राइकोडर्मा पॉलीस्पोरम, अच्छा 3 है। साइक्लोस्पोरिन ए राइट, राइट ए राइट। मोनास्कस परफिडियस स्टैटिन लोअरिंग ब्लड कोलेस्ट्रॉल राइट। स्ट्रेप्टोकॉकस व्हिच वन इज इनकरेक्ट मोना ना कि स्ट्रेप्टोकॉकस स्ट्रेप्टोकाइनस रिमूवल ऑफ क्लॉट राइट। Clostridium botulicum lipase no, কি করবে এটা? butyric acid rating of fibers, jute fibers, rating of fibers বা jute fibers, ঠিক আছে। চল জলদি করো, আমি এটা দেখছি না, অপশনস আমি নট ওয়াচিং, কাম অন ডু ইট। সজিত টেল মি। ऑप्शन नीचे, अच्छा करो, चलो करो। करो D A3 টাইকোস মনোসোডিয়াম প্রোডাকশন অফ ইনসার্ট, ভেরি গুড। এখানেও কি তাই না? 3 দুটো আছে, চল ভালো করে দেখ। So A3, okay, A3. Monascus perfidius for production of blood cholesterol lowering. Trichoderma immunosuppressive. Propionus pharmacomani switch chains. What answer? D2. D2. Yes, answer. D is the answer. This is the answer? Sure. Now see. A3, B1, B. Okay, done. D is the answer. Okay. Good producer of citric acid is Pushpita. Good producer of citric acid is citric acid. Aspergillus niger. Aspergillus niger. Now, tell me, who is going to tell me about Pseudomonas? Huh? Very good. Putrefying bacteria. It can feed on the oils. It can feed on the petroleum products. Also, Pseudomonas is important for denitrificants. Denitrifying bacteria. Denitrifying bacteria, Pseudomonas. What is the other term for that? What is the other example? Thiobacillus. Clostridium botulinum, you know. Saccharomyces, you know. Aspergillus answer. Answer is Aspergillus. Monascus purpureus, Sojit, is a yeast used commercially in the production of Streptococcus purpureus clots. Monascus purpureus. Okay.
Sir, option B.
Oh, Monascus. Sorry, blood cholesterol lowering agent. This is about Streptococcus. Blood cholesterol lowering agent. Hoche Monascus purpureus. Chalo, Rupkotha, you tell me. Next one. A patient brought to a hospital with myocardial infarction is normally immediately given by good blood cholesterol, clot buster. Yeast is used in the production of bread and beer. Bread and beer. কি সুন্দর সাজিয়েছে দেখ bread and butter হয় bread and beer cheese and butter দিয়েছে okay চলো read the following statements to blanks. A drug used for dash patient is obtained from the species of the organisms. The one correct option for two blanks is ठीक है. A drug used for organ transplant. A drug used for organ transplantation patient is obtained from a species of the organisms Trichoderma polysporum. B is answer. তাই তো হার্টের সাথে পেনিসিলিয়ামের কোনো সম্পর্ক নেই, সোয়াইন ফ্লুর সাথে মোনাসকাসের কোনো সম্পর্ক নেই, AIDS has got nothing to do with Pseudomonas. Yes, Shojit. Which one of the following is wrong matching of microbes and its industrial product, while the remaining these are three are correct? আচ্ছা, ওরে বাবা, হয়ে গেছে, পাওয়া গেছে দুষ্টু পাওয়া গেছে. দুষ্টু is A. Yeast statin. Which of the following statement about methanogen is not correct? Not correct. Mind you, whenever you get this kind of questions, you should take out the pencil and mark over here. Not correct. They produce methane gas. They can be used to produce biogas. They are found in the rumen of cattle and their etc. They grow aerobically and break down cellulose-rich food. What is the answer? They grow aerobically and break down cellulose-rich food. কি হবে? Anaerobic digester. The guts of cow and buffalo possess. Tell me about chlorella. What do you know about chlorella? What is chlorella? Bolo. Green algae. Helps in
space astronauts.
Space travelers. They take chlorella because they can produce what? They can help in photosynthesis and can produce oxygen. Number one. One kind of antibiotic can be produced by them. Chlorellin. Methanogens, you know. Cyanobacteria. Cyanobacteria, number at example of cyanobacteria. Nostoc, good. Aramina, nostoc. Any other one? Eulothrix, oscillatoria, oscillatoria. Fucus. What is important about fucus? Is a brown algae. Fucus is a brown algae. It contains algin. It contains algin. Fucus. In gobar gas, the maximum amount is that of methane. What percentage? 70. Rest 25% carbon dioxide. Rest 1% hydrogen sulfide and hydrogen. Gobar gas contains CH4 and CH2. A. এটা বিওডি পড়াইনি ছেড়ে দে following is protein alarming large digester বলবো না we have not done this chalo next day we are going to talk about all this clear no we have not done this এটা লাস্ট এটা পারবো না হচ্ছে বল for rating of jute rating answer is butyric acid bacteria butyric acid by Clostridium butylicum Clostridium butyl tell me where can you find this Helicobacter pylori inside your stomach Helicobacter pylori more of it more the acid content Streptococcus lactin methophilic bacteria biofertilizer we have not studied so next day we are going to also solve all this. That's all about microbes in human welfare for today. Next day we are going to complete STP, sewage treatment plant, and biofertilizer and biocontrol agents, and we are going to finish off this chapter. And we are going to solve some more questions from here. Clear? That's all for now. Sure.